So we are into the pharmacodynamic system, and we have already learned and discussed about the different types of receptors and the way which receptor works. Now important aspect after that is the receptor regulation, how this receptor gets regulated. Now if you see this receptor regulation thing, there are two basic terminologies: receptor desensitizations and receptor down regulations. Now we need to understand what this means, what this means by down regulations and desensitizations. Now when this agonist molecule gets exposed to the receptors for longer time, the eventual response decreases. There is decrease in the response. Why? Because of the constant sensitizations or the constant exposure of agonist to the receptor. So because of the constant exposure, the response of the receptor decreases. Now this decreased response occurs due to two mechanisms. One is down regulations and one is desensitizations. So what do you understand by down regulation? Down regulation means decrease in the number of receptors. So receptor number decreases. So what is the mechanism? How this receptor number decreases? This receptor gets internalized. This receptor gets internalized, goes inside the cell. So internalization of receptors, and these internalized receptors eventually gets destroyed by different lysosomes. So because of these lysosomal actions, these internalized receptors gets destroyed, and due to which there is decrease in the number of receptors, and due to which there is decrease in the response, and this process is called as down regulations. Now what is desensitization on the other hand? Desensitizations means decrease response, but here the mechanism of decrease response may be different. Here the mechanism is at receptor level. Okay, so any defect at the receptor level produces desensitizations. Now which kind of defects? This defect may be receptor inactivation. This receptor gets inactivated, or there is faulty signal processing. The signaling process is faulty, or there is some inhibitory protein formation is there, and which interferes with this signal processing process. So any defect at this receptor level due to inactivations or because of any faulty signaling protein or inhibitory protein formations, there is decrease in the response, and that is known as desensitization. So hope you are clear that decrease in the response is because of two mechanisms: down regulations and desensitizations. Down regulation is decreased number of receptors, and desensitization is defect at the receptor level. Now if this decreased response occurs quickly, that is known as tachyphylaxis, and if this decreased response is gradual, that is known as tolerance. So these are the two terminologies we just need to remember in brief. What is tachyphylaxis and tolerance? Tachyphylaxis is basically decreased response in a quick session. Like for an example, we have given drug here and there is normal response. Again we are giving drugs, there is decrease in the response. Again we are giving drugs, there is decrease in the response. Again we are introducing drug and there is decrease in the response. So quickly the response decreases, and this process is known as tachyphylaxis. While what happens in tolerance, there is decrease in the response, but this process is gradual over the longer period of time. So if over the longer period of time response decreases, that is called as tolerance, and if there is quickly decrease in the response, that is called as tachyphylaxis. How this happens, what is the mechanism of this tachyphylaxis in tolerance, we'll discuss later on. So the other aspect in receptor regulation is upregulation of the receptors and supersensitivity of the receptors, and it is quite opposite of down regulations and desensitization. Here in upregulation, there is increase in the number of receptors, as you can see here. So increase in the number of receptors, and this occurs because of deprivation of these receptors from endogenous molecules or agonist. So if this receptor gets deprived of its ligand or the agonist, it produces reflexly increase in the number of receptors, and this is called as upregulations. What is supersensitivity? Increased sensitivity of the receptor. So here, if the sensitivity of the receptor increases, it is called as supersensitivity, and the mechanism is again kind of a same. If the receptors is been deprived of all the agonist for longer period of time, and here in this case clinically any nerve injury for an example, so in this case the receptor becomes more sensitive, and now if any agonist gets exposed to it, it produces exaggerated response. So example of it is clonidine withdrawal. Okay, clonidine is a drug basically, and it is being used as an antihypertensive agent, and we'll see in ANS that patient is taking clonidine and suddenly it stops, the blood pressure shoots up and the response is exaggerated. So there are different clinical scenarios in which these upregulations and supersensitivities are also involved. Similar way, if antipsychotic drugs, those are D2 receptor blockers, are given for a longer time, then these receptors becomes super sensitive, and now if any of these agonists gets exposed, there is exaggerated response is produced. So these are the clinical scenarios we need to keep in mind about upregulations and supersensitivity of receptors. Important aspect in this pharmacodynamic is dose response curve. Let's try and understand this figure. Now what this figure is elaborating, here on x-axis it's showing increase in the dose, and on y-axis there is response. So it's a dose response, mainly the relationship. Now if you increase the dose, there is gradually increase in the response, right? So this is how we get this dose response curve, and normally this dose response curve is hyperbola kind of curve. But what is the limitation of this kind of dose response curve? On this dose response curve, we cannot plot many responses. So there are only limited responses we can keep in this dose response curve. For more responses, if we want to get more responses, we need to convert this dose into the log dose. So now if we get log dose here, we can plot more responses in comparison to the normal dose, and the curve becomes more of a sigmoidal shape. So what is the inference, or what are the advantages of this log dose? Is wide ranges of doses can be plot, and second, here the mid portion is quite linear, so we can establish the relationship directly. And third important thing, we can compare these responses of different drugs as well. So that becomes easy to study agonist and antagonist. So these are advantages of log dose response curve over normal dose response curve. Here we need to understand some of the definitions or the concepts related to those dose response curve. Let's understand what is potency or a drug potency. It refers to the amount of the drug needed to produce certain response. So if the less amount is needed to produce a certain response, that is more potent, and if more amount of drug needed to produce a response, that is less potent. Quite understandable. So on this log dose response curve, potency is more on the left side. Okay, so potency is more on the left side. If dose response curve is more on the left side, that drug is more potent, you can say. Yes. What about efficacy? efficacy, we have already seen efficacy in the receptors, but here in relation to dose response curve, efficacy is the ability of a drug to produce maximum response. So it is more related to height of the graph. Yes, so if the log dose response curve has more height, the drug is more efficacious. If the dose response curve is more on the left side, the drug is more potent. What is the important out of these two, drug potency or efficacy? So clinically, efficacy is the thing that we need to keep in mind while choosing different drugs. Here an example is been given where there is four drug A, B, C and D. Graph is given. All the drug is been given in the form of log dose response curve. So response of all the drugs is been plotted in a single graph, and now if we want to compare, what is the inference we can get from comparisons? Now, which are the highly efficacious drug out of these four? Drug A and drug B. Why? Because efficacy is more related to height. So if they produces maximum response, that is more efficacious. But which is more potent out of this A and B? A, because A is more on a lefter side, so that is why A is more potent in comparison to drug B. But what about drug D in comparison to drug A, which is more potent? So yes, drug D is more potent in comparison to A, but drug A is more efficacious in comparison to drug D. So this is how you can compare different drugs, and depending upon that, you might be asked in examinations. With that also we need to understand slope. As we have seen in log dose response curve, we get a middle portion quite a linear, and that is called as slope, which is quite steeper. Okay, but still some of the drug having the log dose response curve are quite linear or quite steeper in comparison to other drugs which is more of a curved. So here two drugs been described. One drug is hydralazine and one drug is hydrochlorothiazide. So let's compare these both. What these drugs are doing, the response is fall in BP. So both the drug decreases the blood pressure. So what if we increase the dose? If we increase the dose of this hydrochlorothiazide, there is increase in the response, but not that great. But what about this hydralazine? If we increase the dose, there is constant increase in the response. So here the log dose response curve of hydralazine is quite steeper, and so if we increase slight dose, this will create increase in the response. So slight increase in the dose create dramatic increase in the response in case of hydralazine, while slight increase in the dose won't affect much into the response in case of hydrochlorothiazide. And this we can decide on the basis of slope, or how this drug dose response curve of that particular drug is. Another important terminology with that dose response curve is therapeutic index. Now therapeutic index, as the name suggests, it's a measure of safety, and many a time this question is asked as an MCQ. What therapeutic index basically is showing? So it's basically showing a safety of a drug. So therapeutic index is for safety of a drugs. In this figure you can say there are two drugs which are red color drug. So this is red color drug and this is blue color drug. There are two drugs there. Now this red color drug having two response. This is response A and response D, and this blue color drug having its two response. This is response B and response C. So the gap between the two responses of the same drug is more. So this gap is called as therapeutic index. So for an example, let's say this red drug is beta agonist. We'll see in ANS section. So this beta agonist produces bronchodilation. So this graph is of bronchodilatations, and this graph is of cardiac stimulations, or you can say side effects. So there are two graphs. Graph A is of use and graph D is of side effects. So if the gap between its therapeutic effects and its side effects is more, then it is called its more therapeutic index. So this gap is called as therapeutic index. While in blue drug, while this blue drug having very little gap between the two responses of side effects and its therapeutic use. So it's called as narrow therapeutic index. That is called as narrow therapeutic index. So drugs with high therapeutic index, are quite safer. So if the therapeutic index is high, these are safe drugs. And if the therapeutic index is very narrow, these are not safe. So they are not safe. They are very dangerous. And we need to maintain the concentrations accurately. In practical scenario, in experimental things, we calculate this therapeutic index on the basis of LD50 and ED50. Now what is LD50? That is lethal dose 50. And what is ED50? That is effective dose 50. Lethal dose 50 means dose which is lethal to the 50% of animals. Okay, or if we have taken 100% of animal in any experimental study, and on a particular dose where 50% of animal dies, which is lethal, that dose is called as lethal dose. And ED50 is effective dose for 50% of the population of the experimental study. So we get this LD50 and ED50, and on this formula we can get the value of therapeutic index. If the value of therapeutic index is high, the drug is safe, and if the value of therapeutic index is low, the drug is more of a toxic. So this is a clinical inference of therapeutic index. Now next thing is therapeutic window. Now what is it? For an example, in earlier part also we have seen that drug is having its two effects. One is therapeutic effects, and still if we increase the dose, it produces adverse effect. So the point between minimal therapeutic effects, the point from which the therapeutic effect starts, and the point of maximal acceptable adverse drug reaction. So maximum side effects which is acceptable. So the area between these two is called as therapeutic range. So it's a dose range basically. So it's a dose range between the 2 points, minimum therapeutic effects and maximal acceptable adverse drug reactions. After which, if we still increase the dose, there are tremendous side effects which are not acceptable, which are dangerous. And below this point, there is no therapeutic effects. So this window is called as therapeutic range. So this is also called as therapeutic window or therapeutic range. So sometimes the drug which is been given in a form in certain books in therapeutic range, for an example 8 mg to 16 mg. Now what it says, this is a therapeutic range that says if we give less than 8 mg, that won't be any effect, and if we give more than 16 mg, there are chances of side effects. So this is called as therapeutic range, and we need to understand the drugs which are having high range and drug which are having low range. So high therapeutic index, low therapeutic index, or high therapeutic window and low therapeutic window, we need to understand. So drugs like phenytoin, that we have already seen, drugs which low therapeutic window. So phenytoin, drugs like warfarin, like these are certain drugs like theophylline. So these are drugs, these are some of the examples we have already seen in TDM chapter. So these are the drugs that we need to, they are having a low therapeutic window or the narrow therapeutic window or the narrow therapeutic range. So their concentration range, fluctuation concentration range is very narrow, and that is why we need to keep an eye on their concentrations. Another aspect of this dose response curve is drug selectivity and drug specificity. So selectivity is more related to action of a drug, how selective that drug is to produce its action, and specificity deals with its receptor, how specifically it acts on a receptors. So if drugs acts on, for an example, both the receptors beta 1 and beta 2, whether it is having high specificity? No. If drugs acts on particular receptors only, like for an example beta 1, so it is more specific, right? So these things you need to keep in mind, selectivity and specificity. Here in this graph there are two drugs, red drug and a blue drug. Red drug having two effects and blue drugs having two effects. Now this red drug you can say is more selective because the effects are quite wider, while this blue drug is less selective because the therapeutic effects and the side effect ranges are quite narrower. So on that basis also you can decide the drug is selective or not.